Hey guys, this is Dr. L, and this is the pre-lab talk for our yeast metabolism lab. There were two goals in this lab. Both of them were intended to reinforce your hopefully growing knowledge about cell bioenergetics. In other words, how cells can metabolize things to make ATP. The first part of the lab was asking the question, what sugars can the yeast Saccharomyces cerevisiae metabolize? And the second part of the lab was asking the question, how can we use the methylene blue viability assay to determine if Saccharomyces cerevisiae are alive and metabolically active? So first, a little bit about S. cerevisiae. It is here's a picture of it. So this is an electron microscope. An electron microscope is used to capture this image, and the credits down there on the bottom. And this shows Saccharomyces cerevisiae, which is otherwise known just as common bread yeast. We use it to make bread, and we use it to make wine, and we use it to make beer. We use it for a lot of different foods and beverages. It's capable of ethanolic fermentation, and it also can produce gas, a lot of gas, which is what leavens leavens bread, raises bread, and other baked products. So we like to use it in the kitchen. We also use it as a model organism to better understand eukaryotic cells. This is a eukaryotic microbe, and we can study all sorts of different things, including cellular division using S. cerevisiae. So it's a very important model organism, easy to work with, easy to grow, wealth of information and knowledge to be gained by studying it. And it's used industrially to do fermentations of industrial relevance, things involved in the pharmaceutical or chemical industries, for example. This yeast can reproduce asexually through the process of budding, and this electron micrograph shows an S. cerevisiae cell budding. Looks like a little tiny snowman, but that's a budding cell, mother cell and daughter cell. Or during times of stress such as starvation, this yeast exists in two mating types, A and alpha, and those organisms can produce spores, and those spores can unite and participate in the process of sexual reproduction. So it's a really interesting organism. Now back to the metabolism side of things. So this is a really messy drawing, and this is what I had up on the screen or up on the blackboard when I was doing this live with some of you guys, but you should recognize parts of this sugar. So if we're thinking about the kinds of metabolism that can happen in yeast, they are capable of aerobic respiration, and they are also capable of what is called ethanolic fermentation. So let's cover the first one, aerobic respiration. And aerobic respiration is sugar, usually glucose, but it could be other sugars too, and that's what we're testing, is metabolized through the glycolytic pathway to a molecule, actually two molecules of pyruvate. And that conversion from sugar, from glucose to pyruvate, is done by what is called glycolysis, or the glycolytic pathway. It constitutes a sequential process by which electrons are removed from the original sugar molecule, and they are placed on the coenzyme NAD+, which reduces it to its reduced form NADH. The pyruvate is then able to, after a little bit of preparatory reaction, is able to enter a cycle called the Krebs cycle, also known as the TCA cycle or the citric acid cycle. And in the Krebs cycle, the pyruvate is successively oxidized so that all of the available electrons are removed from its structure, releasing carbon dioxide. The electrons that are removed are used to reduce NAD+ and FAD, an additional coenzyme. Those molecules are converted to their reduced forms, NADH and FADH2, and those reduced coenzymes are able to take the electrons off to the inner mitochondrial membrane, where they drop them off at a structure known as the electron transport chain. So at the electron transport chain, what happens is these reduced coenzymes are going to drop off their electrons to protein complexes embedded in the inner mitochondrial membrane. I've drawn two blue structures. These are the protein complexes. There are actually more. This is a simplified drawing. Electrons are being received and then transmitted through this electron transport chain, which includes protein complexes outlined in blue, and then this little purple thing is supposed to represent a quinone molecule, which are also parts of the electron transport chains. As the electron transport chain transfers electrons from place to place, it is simultaneously able to pump protons, which are H+, through these protein complexes, causing a proton accumulation on one side of the inner mitochondrial membrane. That is known as the proton motive force. More on that in a moment. The electrons at the end of the day are going to end up being dropped off onto oxygen, which is the terminal electron acceptor, thereby reducing it to water. And that's the end of the road for the electrons in this little story. Now, this proton motive force serves to power the synthesis of ATP by a really key protein, which is shown in dark blue here. That protein's name is ATP synthase. ATP synthase, very, very important protein. It does two things. It makes ATP in a process known as oxidative phosphorylation, but it powers that production by serving as a channel to allow the protons, which are kind of all clustered on one side of the inner mitochondrial membrane, it allows them to flow through only ATP synthase's channel back to the other side. So these protons are concentrated here. That's an electrochemical gradient, again called the proton motive force, and the only way they can spread themselves out is flowing through this ATP synthase channel, and they will do that, and that flow of protons will power oxidative phosphorylation to generate ATP. Okay, so back over here to the glycolysis and the Krebs cycle side of things. Okay, so an outcome of the Krebs cycle, an output of the Krebs cycle, is the molecule CO2. And CO2 in a liquid will show up as a gas bubble. So if you see CO2 in a solution of yeast cells doing this process, you should see bubbles. Now, if the cells run out of oxygen, then everything over here on the right-hand side will halt, and actually so will the Krebs cycle. But what will happen is cells will switch to the process of ethanolic fermentation. And in ethanolic fermentation, which yeast are capable of doing, a sugar is metabolized by glycolysis to pyruvate, but then that pyruvate, instead of entering the Krebs cycle, is fermented to the molecules ethanol and CO2. And along the way, ATP is produced. And so whether yeast are doing full-on aerobic metabolism and running their Krebs cycles, or doing ethanolic fermentation, in either case, gas bubbles which contain CO2 are going to be produced. So bubbles are always evidence of metabolism. If you feed yeast sugar, and they have the other things they need in their environment, and if you see bubbling, then they are metabolizing that sugar one way or the other. You could be more sure about that if you did things like measured the oxygen concentration. If there was no oxygen, you'd know it was ethanolic fermentation. Or you could measure directly for ethanol production as well. You can also smell ethanol, so you could tell if they were doing that. So in our lab, we set up a triplicate experiment where we tested 3 different sugars and we included a no sugar control. We mixed the yeast plus some appropriate buffer plus the different forms of sugar, glucose, sucrose, or lactose. And we did that in triplicate. That's what the ×3 means. And we monitored for the production of bubbles. If there were bubbles produced, then we knew that there was metabolism. If there were no bubbles produced, we scored them as negative, no metabolism. You can see another video in this series to see what that actually looks like. The second part of the lab was the yeast viability assay. And this takes advantage of the inherent properties of a chemical called methylene blue, which is shown here in the top. When methylene blue is in its oxidized state, it is blue. If it gains electrons through the process of reduction, then it will be reduced, and then it will form a different chemical called leucomethylene blue, which is colorless. So if you see blue, it's oxidized. If you see no color, it's reduced. The electrons that methylene blue gains come from metabolic reactions. And so this is a really common assay used in a number of different contexts. It's It's used in the brewing industry to see if yeast are alive or not, and it's used in the dairy industry to see if there are bacteria inside of milk, which could cause the milk to spoil. And that's what the picture is showing. So this tube here that's completely blue has it's got milk. All three tubes have milk in them. The blue one has no bacteria in it, and so there's no metabolic activity, so it stays blue because there's no release of electrons to reduce the methylene blue. This has got a little bit of bacteria in it, so we're seeing it like go a little bit towards colorless. And then here we've got completely colorless milk, which shows that there are a lot of bacteria in it, and that milk, those bacteria are doing metabolism, freeing up electrons, which are being placed onto the oxidized form of methylene blue, reducing it so that it goes colorless. In the brewing industry, you wouldn't want to set up a batch of whoops, sorry about that, guys, of beer making, with dead yeast. So it's very common to take a look through the microscope and stain the yeast that you have. Why is that? Stain the yeast that you have to see if they show up as blue or colorless. So this is a picture that Katie shared from lab, and you can see there's some blue cells and there's some colorless cells. The blue ones are dead, the clear ones are alive. And the same is true for Lindsay's image shown here. And then this is a more finely focused image using 100x objective from a research paper that I've given the citation from here. So that's kind of the backdrop or pre-lab talk for this lab. Take a look at the other videos to see what else happens.